welcome to another Piper Pod. This time we're going to focus purely on that adaptive immunity. Looking at this picture, what we've covered so far is an introduction to the natural killer cell, which is directly on the left. So this lesson, we're going to learn everything else that's in this picture. So stay tuned and I hope you enjoy it. When we talk about adaptive immunity, we are starting to look at the absolute specifics of immunity. So innate immunity is non-specific, so it just kills anything it doesn't recognize as self, or it's a natural protective barrier, and that's how the first and second lines of defense work. When we start moving to the third line of defense, we start looking at immunity, as in having immunity for a specific antigen or a specific threat. Now, the adaptive aspect of immunity is covered by lymphocyte cells, okay, the other half of the lymphocytic cell growth. Now, the way lymphocytes work is they have the ability to register a specific antigen, hence the specific immune system. Now, an antigen is a substance that elicits an immune response. Now, what that means is, it's kind of obvious when you say it like that. It means that something is registered in the body that is identified as non-self. Based on our genetic growth and our development, every one of our cells has a chemical tag that identifies us as self-growth. Now, obviously, there's certain things in the body that are not exactly the same as our genetic material. Okay, there's obviously the cells that don't have any genetic material, like red blood cells, but as we've already heard, they get a self replicator put on them, and they're only really subject to certain types of antibodies that are produced. And other things like gonad development, ovums and sperms, they're protected by innate body defenses, same as childbirth. Okay, but in order for every other non-self antigen to be picked up, an antigen, in order to cause a problem in the body, it has to be large enough. And we'll learn about different types of antigens soon, but most of the ones are usually associated with some form of complex structure. Okay, so it's not a simple element or something like that. It has some form of complex structure to it, and if it wasn't for the size and the complexity, then it wouldn't be recognized as foreign. It would just be recognized as one of the particles or one of the electrolytes that we absorb. Now, looking further into an antigen itself before we move into the immunity of them, you have to understand that there's different types of threats. Okay, so the most common one is what's known as a complete antigen. Now, a complete antigen is a standalone, one antigen elicits a response from one antibody, and then it's fine. Okay, normal things like foods, pathogens, they often have one antigen that elicits some form of response. Okay, if we start moving into epitopes, then what that means is it's a large enough structure to have multiple different recognizable antigens with it. So this one epitope can have numerous antibodies formed against it. Okay, so it makes it a little bit different. And then when we start looking at things like drug allergies and things like that, or most common one is penicillin, then we start looking at incomplete antigens. So by itself, a hapten is a really small foreign particle, okay, and it will not elicit an immune response unless it's piggybacked onto a larger molecule. Okay, so if it binds with the blood, then all of a sudden it can be seen as a true antigen and therefore elicit the immune response. By itself, it float around the body completely harmless, going undetected. Okay, so that's the different types of antigens that are out there. So every time I refer to an antigen, I mean some form of foreign threat. When we look at the third line of defense, we are looking at what's known as adaptive immunity. Now, adaptive immunity is broken up into two types of immune responses. And by the end of it, you'll understand why we need two different types, because they both have their unique function. So we have what's known as the humoral or antibody-mediated immunity, and that is all associated with B cells and their production of antibodies. And then we have cell cell-mediated immunity. And that is looking at specialized T cells, a version of the lymphocytes, that have the ability to attack viruses or tumor cells. And we'll talk a lot more about them as well. Now, in order for any of these two types of immunity to happen, they all start pretty much the same way, or they have to have some involvement with a cell known as the antigen-presenting cell. So as an added bonus, I'm going to include information about the antigen-presenting cell, so that way we'll be able to draw links with the innate immune system and realize that these two immune systems work hand in hand together. They are not two separate systems that work independently of one another. So let's start the story right at the beginning, which is obviously with the production of these specialized lymphocytes. Now, in the first presentation on AMP, we went through the blood. So you already know how lymphocytes are formed. Okay, these were the special ones that come from the lymphoid division. Okay, lymphocyte, lymphoid, it makes sense. And then from there, they get divided into initially B cells and T cells. Okay, now T cells, for starters, they move out of the bone marrow. Okay, so they migrate out of the bone marrow, and initially when they move out of the bone marrow, they're what's known as immature T cells. Okay, so they're not immunocompetent at all. They migrate to the thalamus gland. Within the thalamus gland, they go through what could only be described as rigorous induction and training processes. Okay, so they're put under a huge amounts of stress. And any T cell that doesn't quite make the cut is destroyed. Okay, now what do I mean by not making the cut? Obviously, I'm only using analogies like this so it makes it easier to understand. But if there is a T cell that in its processes recognizes self instead of non-self, it's identified in the thalamus and it is destroyed in the thalamus. Okay, from there, it goes through its specialized function, so it can become a certain type of T cell. Okay, it is also subject to further development, which is similar to the B cell. So we'll leave the further development until the end before the migration to the lymphocyte. Now, B cells, they stay in the bone marrow. So it's nice and easy to remember. B cell, bone marrow, T cells, thalamus. Okay, nice and simple. Now, B cells are also subject to similar sort of stresses. They're weeded out. They're kept developing in the bone marrow until they're ready. Any B cell that sneaks out of the bone marrow before it's ready will be destroyed as a non-self. Okay. Now, whilst each one of these immature or immunocompetent, naive, whatever you want to call them, cells is in the bone marrow or the thalamus, they are built with different sorts of receptor sites on the outside of their cells. So the receptor sites on the outside of their cells are unique in order to register different antigens. Okay, so that material comes from our genetics. Okay, that's why certain genetics are more susceptible to certain diseases than others. Okay, because your genetics determine not your immunity, but it determines the receptor sites that are found on your immune cells. So your lymphocytic cells will have certain and different immune receptor sites on the outside of them. Now, there is estimated, obviously unknown, to be around 1 billion different receptor sites found on these cells. Okay, 1 billion different ones. They have 2,500 different base nucleotides that they can choose from. So with that 2,500 different combinations, they can make up to 1 billion different receptor sites. And the reason that's handy is because it gives us a wide and diverse ability to register different antigens in order to elicit the immune response. Once the B and the T cell are ready, they're what's known as naive lymphocytes, as in they have not been registered, they have not picked up their threat, they have not been bound to their antigen. So they move to the lymph nodes and the lymphoid tissue in order to wait for the antigen to come along or be delivered. Now, what I mean by delivered, that is where our antigen presenting cell comes into play. Okay, so macrophages or dendritic cells play the main role when we're dealing with antigen presenting cells. Now sometimes already matured B cells can be the initial cell that delivers the antigen. But exactly as the name of the cell sounds, the antigen presenting cell, would have you believe, that's exactly what it does. When these cells, macrophages or dendritic cells, go through phagocytosis, they break the threat down and they then present the antigen on the outside of their cell. From then they migrate to the lymphoid organ and they try to find a helper T cell with the right combination. Okay, so imagine the antigen presenting cell as somebody who has massive intel on a threat, and it goes to the lymphoid organs in order to find somebody, in this case a helper T cell, who will listen, who is actually going to take their threat as a credible source. Once that credible source is found, the T cell will bind to it and then release a bunch of chemicals, and those chemicals that release is the starting point of our adaptive immunity. Okay, and on top of that, it also, if you will, ordains the macrophages in particular to go through to basically upgrade. And all of a sudden the macrophages become insatiably hungry for pathogens. So they're incredibly phagocytic. And at the same time they also secrete bactericidal chemicals, will roam through the body and kill any bacteria that it finds. This has all happened because the helper T cell picked up the antigen as a credible source and started the whole process. Now this starting point is both needed, it's one way or another, in both your humoral and your cell-mediated immunity. So let's start looking specifically at the two different types of immunities. When we look at humoral or antibody-mediated immunity, it involves the B cells. Now the B cells have already been matured in the bone marrow and then sent to the lymph organs. Okay, from there the B cells have the ability to pick up an antigen as a free-flowing antigen or get the antigen delivered by the helper T cell. Okay, it is open for both situations. Okay? But what is absolutely needed every time in order for the B cell formation and antibodies to be produced is the B cell needs to be triggered by the cytokines released by the helper T cells. Okay, now I understand that is confusing. Okay, now in this case, this is a macrophage displaying the antigen. So in the bottom, you see the large macrophage going through phagocytosis and then presenting an antigen on the outside of its cell. From there, the helper T cell will come along and it will register the antigen. From there, it's the helper T cell's job to activate the rest of the immune response. Okay, so from there, it has a choice. It can either travel to a B cell and present the antigen to them, or if the B cell has already registered the same antigen, it releases cytokines in order to stimulate B cell proliferation. Now, B cell proliferation means once the individual B cell is activated by cytokines and the antigen, it makes what's known as clone B cells. Okay, and from the clone B cells, they produce more proliferation and differentiation. They mostly become what's known as plasma cells, but then a few of them become memory B cells. Okay? Now the plasma cells, they're the ones responsible for the production of antibodies, and they produce a lot of antibodies. Okay, so we're talking 2,000 antibodies a second can be produced by activated plasma cells. Okay, so that is a phenomenal amount of antibodies that has been pumped out into the system. The memory cells at this stage don't do anything. Okay, they're like your intel gatherers. They're not fighting the fight. They're storing the information. They're storing the history of the antigen attack, So then next time the antigen attacks, they can be activated. Now, people often talk about immunology, okay, and they often talk about the difference between a primary response and a secondary response. So this picture here will be pretty similar. Okay, so that entire process has just gone through, and it obviously ended up in the production of plasma cells producing antibodies. But then we also have a memory cell. So all of us would have been told in the past that that means that now that we have a memory cell, if we're exposed to a same antigen, that memory cell was activated straight away from the antigen and go through proliferation, producing plasma cells and memory cells straight away and producing antibodies in order to take care of the problem. Now, when you talk about it in a linear fashion like this, both sound really quick. So the secondary response only really sounds marginally faster. But the difference is, primary response before the production of antibodies can be a matter of days. Okay, 1 to 2 days in order to produce antibodies. Now, in the secondary response, the production of antibodies can occur within 1 to 2 hours. So that is a massive difference. And most of the time, the secondary response will have pushed out antibodies and dealt with the problem before you actually feel sick. As opposed to the primary response, the first time you get sick, you'll have symptoms because it's 1 to 2 days later. Okay, just remember how long it takes back in that innate phase for macrophages to be activated, 12 hours, engulfing through phagocytosis, then traveling to the lymphoid cells, finding a T cell that's actually going to listen, that T cell releasing the chemicals to the B cell to start proliferation, and then cellular division starts. Okay, long process. Secondary response, the memory cell floats around the bloodstream. It's chemically attracted to that antigen. As soon as that antigen enters the bloodstream, it's drawn to it. It binds with it, starts proliferation straight away. A hell of a lot faster. Okay, so that's the difference when you're talking about memory and the importance of why memory B cells can really help us out when dealing with fighting pathogens. Now, the production of antibodies can happen a number of different ways, and essentially your body doesn't care how it gets the trigger. So it doesn't care how the antibodies get formed, as long as they get formed. The process is similar. So we have two different pathways, which is active versus passive. Now, your active method can either be naturally or artificially acquired. Naturally acquired is the process we just spoke about. Okay, the very long, drawn-out process of being exposed to a pathogen, finding that antigen, and producing antibodies. Okay, now vaccinations also is an active way of getting immunity because it's not putting in antibodies, it's putting in an innate version of the antigen or the pathogen itself, and therefore it will produce antibodies through it. Now, the reason you need boosters and why vaccinations can sometimes not last as long as your naturally acquired immunity, which can be for life, okay, is because the artificial antigen triggers directly to the helper T cell. Okay, so it does not involve the initial side of the house. It does not involve the initial innate response. Okay, so we don't go through the innate growth. And believe it or not, that plays a big role in how long the memory cells are actually going to hang around for. Okay, without the communication with the dendritic or the macrophage, the memory B cells will actually not hang around as long. The other version of humoral immunity is passive. Now, naturally acquired is pretty much associated with babies. Okay, so fetal development, any antibodies can be passed through the placenta. So the baby will be born with the antibodies flowing through its body. Okay, but it won't have any memory cells, and it won't have any activated or any B cells that's associated with picking up any antigen. Okay, it can get additional antibodies from breast milk, but again, these are short-lived. As to the artificially acquired, So literally your tetrotoxins, your gamma globulin injections that are used to fight certain disorders or help certain disorders, they're artificially acquired passively. Again, they do not last as long because there is no cellular memory associated with the passive process. It's just a boost, if you will, in order to aid in the whole immune response. Now, when we look at what's being produced, okay, the end result of all this, what we've been talking about, the end result of the humoral development or the antibody immune response is the production of antibodies. Now, antibody is a series of polypeptide chains. Okay, they're usually shaped like a Y, but they can be shaped like a T as well, just for interest. The protein itself is what's known as a gamma globulin, and it is the antibody that is designed to bind to a specific antigen and in turn render that antigen useless or targeted for elimination. Okay, that's what antibodies do. Now, looking at a picture of an antibody, this is a very simple drawing just to help represent what it is and what they look like. Okay, so the variable region of each antibody is specifically designed to bind to an antigen site. So it's designed to be attracted and hold onto different antigen sites. Okay, each of the light and heavy chains in the constant region are really held together by disulfonide chains. Okay, so two sulfur molecules bound together in order to keep its structure. When you're dealing with antibodies, like most things in the body, you are not just dealing with one type of antibodies. When they are flowing around in the body, they are known as immunoglobulins. Okay, so they are globulins that assist in immunity. That's what they do. Now, each immunoglobulin is associated with a different class and therefore different function of immunity. So there are 5 different classes to antibodies. Okay, and they're each broken up and they're each designed to perform slightly different roles. Now, your IgM is your largest of all of your antibody groups. Okay, it is what's known as a pentamer. Now, what that means is the picture we saw before, that's 1. Okay, that's a monomer. A pentamer means that there's 5 of them stuck together. Okay, now these IgM are the first antibodies to be released when you're dealing with an active attack. Okay, so that makes sense. You have a pentamer, which then means you have more receptor sites on the actual antibody, means more ability to collect antigens faster. So IgM is the first one to be released in an active infectious state. IgA is a monomer, so it's just one chain. Okay, now if you want to get absolutely technical, because I know a few of you out there may be, this technically can be a dimer as well, which means two antibodies joined together. Okay, so don't shoot me, it can be a dimer as well. Now, the important thing about IgA is that they're found in the mucoid membranes. Okay, so they're found within the mucus and they're there to help prevent pathogens from getting into the body. Okay, so once produced, they can sit into the mucus until they're destroyed or utilized and sit there protecting the body from pathogens getting in. IgD, they're monomers as well, single chain, and what they do is they sit on the surface of the B cells and they have that extra assistance in function to find free-flowing antigens or antigens on pathogens. Okay, so they become an additional receptor site to the B cells in order to aid in the function, okay, and the opsonization in the end of all the infectious antigens. Okay. IgG is the one that you'll hear about the most. Okay, it's also a monomer, and this makes up 75 to 85% of your body's your antibodies found in your plasma. Okay, now this has also been known as the memory antibody, because every time we're looking to see if somebody is immune to a certain disease, we take a blood test and we test for IgG for that particular one. Okay, most famous one in the military that we use is IgG for varicella zoster. Okay, we search for the antibody. If it's present, that means that we have immunity and we have stored memory of that particular one. So that's what IgG does. Now IgG is what's essentially your hellraiser. So IgG it is designed to pick up antigens associated with profound histamine responses, so allergies. Okay, and what it does is once it finds that antigen, it goes and it tells and activates mast cells and basophils to release histamine. Okay, so in highly allergic states, you will have a higher level of IgE antibodies. Okay, so that's the different classes of antibodies that you'll find throughout the body. As you can see, each a slightly different role and a slightly different formation as well. Okay, so that's why we need so many. Like the other cells in the body, each of them have a way to counteract and aid in the overall defense. Now, what antibodies do? Okay, we sort of get the idea that they bind to receptor sites of the antigen. Okay, but how that helps in the overall process is first stage. So antibodies aren't really associated with destroying things themselves. They aid and enhance in other functions that are used to destroy. Okay, so the first thing that they can do is they can basically make certain antigens inactive, okay, by either neutralization. Now what that means is the antibodies will form around toxins and dangerous byproducts of bacteria and viruses. Okay, and they'll bind around it, making it impossible for that exotoxin or virus to penetrate a cell or cause a reaction. Anglination is another thing that they can do that we spoke about when we were dealing with your blood groups, your anti-B, your anti-A, binds to receptor sites, antigen receptor sites on cells and clumps all the cellular masses together. Okay. Precipitation is a similar process, but this is dealing with single soluble antigens and not associated with being connected to a cell. Now each one of those inactivations lead to easier and enhanced phagocytosis and an additional inflammatory response. Now remember that inflammatory response creates a chemotrax and a histamine or vasodilative response, which brings more phagocytes to it that can clean up the area faster. So antibodies aid in that function. The other things that antibodies do is they can enhance the complement protein function that we spoke about in the first lesson. By binding to the outside of the cell, they can actually attract complement proteins to lead to phagocytosis, inflammatory response, and cell lysis. So that's the way that antibodies will work. Okay, the last type of immunity that we have is what's known as a cell-mediated immunity. Now this is all associated with T cells. Now T cells are fantastic at what's known as intracellular antigens. Now certain classes of T cells that we'll introduce, and we'll talk about each of them and the overall process. So the helper T cell we've already discussed. The next major player of T cells is what's known as the cytotoxic T cell or the killer T cell. The others that help the whole overall process is the regulator and memory T cell. So T cell activation happens with the same starting point as B cell activation does. Okay, it all starts with that antigen presenting cell that finds the helper T cell and then starts the process from there. The difference is the helper T cell can't find an antigen itself to activate itself. It needs to have that connection with the antigen presenting cell. It needs the connection so much that it actually needs to bind with the antigen and then be co-stimulated by the APC itself. Now the APC can either stimulate it with cytokines, sometimes, not often, but often it stimulates it with a separate binary site connecting to the helper T cell. Okay, so think of the helper, in this case when we're activating T cells, think of these as your specialized espionage or specialized forces that gather the real intel to find out where things are hiding. So they first need to make sure that the person who's giving the ATP that's giving the information is credible, and then make sure the antigen itself is credible as well. So it needs what's known as double credibility before it will even bother reacting. Once it sees it as a significant threat, or a high enough threat, something that it can deal with, then from there, the APC and the helper T cell release cytokines, and that starts T cell proliferation, T cell division, so growth, and it also helps the bringing of different classes of T cells to help in the overall process. Now, the way T cells different, okay, the main killing aspect is obviously the killer T cells. Now, unlike the B cells that produce antibodies, antibodies can only react to antigens that are found on the surface, either free-floating antigens by themselves or antigens that are on the surface of the cellular or microorganism structure. Cytotoxic T cells can actually detect the antigen of interest inside cellular structure. Okay, so if there is a virus, always a virus inside a cell, bacteria or parasite inside a cell, it can actually destroy that cell. It's also very useful for cancer cells, and this is the cell that's responsible for transplant problems. Okay, so not picking up transplant. The way it works is the cytotoxic T cell will come along, it will land on the outside of the cell of where it's found the antigen, it will punch a hole straight into it using perforins. Perforins will punch a hole straight in the cellular membrane, and from there it will inject lymphotoxins straight into the cell, and that will cause the cell to activate lysomes to self-destruct. From there, cytotoxic T cells are not subject to being over-engulfed and becoming pus like phagocytes or your natural killer cells. That one there will simply finish injecting and then move on to the next cell, and then the next cell, and essentially keep going until they're turned off. So what turns it off? That's where the regulator T cell comes along. Okay, they come along and they dampen the immune response. They tell the C cell, cytotoxic C cells, to stop. Okay, slow down. Basically, no longer required. Everything calm down, and this prevents the cytotoxic T cells mutating and therefore reading different antigens as a threat and having what's known as an autoimmune response. Okay, so if we just let activated cytotoxic T cells through our body when there wasn't a threat, it would cause an autoimmune reaction every single time. Okay, so the regulated T cells calms it all down. Okay, it stops the cytokines being released and stops the process. Now, T cells will generally die within 7 to 30 days. So they can be active in the body 7 to 30 days depending on the threat and the activation of the regular T cells. Now, the memory T cells remain much longer. So they're there hanging around just in case there is a secondary attack from the antigen, or if they've missed a couple of cells that have hidden underneath, haven't matured, come to the surface, that sort of thing, usually associated with ganglions and that stuff. So memory T cells will hang around until they're just so they can find some form of second response. Okay, so there's your classes of T cells. Now, I understand that was a bunch to go through. So hopefully this last slide, in summary, helps the whole process. So just remember that the whole process always starts with an antigen itself. Okay, it always starts with an antigen. From the antigen presenting cell, macrophage or dendritic cell, engulfs it and then finds a helper T cell. Right. From there, the helper T cell activates the B cell and the cytotoxic T cell. From there, B cells produce plasma cells and antibodies, as well as memory B cells. T cells produce cytotoxic T cells and memory T cells, as well as a couple of others. There's obviously your regulatory T cells. From there, the threat is dealt with on two ends. Those antigens that are seen on the surface and the same antigen that is hidden within the cells. On top of that, we also get cellular memory, which means the next time an antigen is presented, we no longer need the presenting cell or the helper T cell. Okay, we just need the memory cells to activate the process again. Okay, so that ends lymphatic system and immunity. Okay, hopefully you got something out of it. Hopefully you now understand that there is a strong connection between the different types of immune systems, and realizing that if it wasn't for the initial inflammatory response stage and simple phagocytosis, then complex specific immunity would never happen. Until next time, take care.